Hello, everyone. I want to welcome you this evening. We're really excited to have this town hall with Ultragenyx here with us and Dr. Heather Lau and to give us a presentation. I'm not going to linger much. I'm just going to welcome each of you, and we're glad you joined us. And I'm going to turn it over to Heather from here.
Thank you, Karen, and thank you to the IFCR for today and inviting us to talk with you all through our Zoom interface, our webinar, to give some research updates from our CDKL5 deficiency disorder research program called UX055. And as Karen mentioned, I'm a physician. My role at Ultragenyx is Executive Director of Global Clinical Development, and I'm the clinical development team lead for UX055. By training, I'm a pediatric neurologist and have been working in academia for 10 years in rare neurogenetic diseases and joined Ultragenyx about a year ago. Next slide. So today's focus, the objective today is to address the CDD community's questions and provide a transparent insight into our 055 program. Questions were submitted by our CDD families to IFCR and have directly shaped our presentation today. So our agenda will start off with a little bit about Ultragenyx for those of you who don't know about us, an overview of our 055 program, including the role of the CDD community in our that have informed our decisions and our plans to date, our overall drug development process, which I think is really critical to understand our approach to drug development, and the status of our CDD research. At the end, we'll have a moderated Q&A. These are based on questions that were submitted already to the IFCR, and I'll be answering them live to address anything that was not addressed during the presentation. Next slide. So who are we and what do we stand for? So Ultragenyx was actually founded in 2010 by Dr. Emil Kakkis. To create a steady pipeline of new and effective therapies and products to treat specifically rare and ultra-rare diseases. I met Dr. Kakkis in 2011 when he first embarked on his initial clinical trial programs in rare diseases, and he worked with my mentor, and that's how I got to know Ultragenyx over the years. It started as a small company of less than 20 people and has grown to over 1,200 employees, with offices both on the West Coast and the East Coast, in global offices in Europe, Latin America, and there's a presence in Canada. But the home base is headquartered in Novato, California. And so Ultragenyx's commitment and Dr. Kakkis's commitment is to develop deep and meaningful engagements with the rare disease community to really understand and address the unmet needs in order to inform a drug development process. Next slide. So like I said before, your interest and questions have directly informed today's webinar. We received 13 questions from the community, and we will answer every question that was submitted. Many of them will be addressed in the presentation, and at the end, we'll have a moderated Q&A where I'll answer additional questions. Next slide. Some of the questions are listed here. These were basically what we found was the community was interested in understanding how a clinical trial will be designed and where it will be conducted, and the potential impacts of a gene therapy. We'll come back to these at the end. Next slide. So successful treatment of patients requires learning directly from patients. One key concept at Ultragenyx about rare disease drug development is that as we learn about rare diseases, we often find that one major well-accepted fact is actually wrong. So what do we mean by that? Well, in medical school, we learn all about different types of diseases. And when we think about rare diseases, it's usually boiled down to some key features. And those features are considered dogma. That's it. However, especially in my academic experience and with the experience of Dr. Kakkis in Ultragenyx, oftentimes when we start to dig deeper, we learn that one of these facts is actually wrong. How do we discover this? Well, it's discovered usually by talking to the patients and caregivers. So direct information is one way to discover those facts and to help us unlock the path to a successful treatment of the disease. For example, as a clinician prior to joining Ultragenyx, I was involved in many different clinical trials in rare diseases. One disease was Morquio, and there was an IV enzyme replacement therapy that was in trial to address bone disease. However, talking to my patients during that trial, I realized that they were having an improvement in their respiratory function, and that it improved significantly over time, that we could see it in long-term follow-up effects with the pulmonologist. Another example, which might be relevant here in the CDD community, is visual impairment. So we were taught, especially by my mentor and the prevailing knowledge at the time, was that visual impairment was actually due to an optic nerve injury, so damage to that optic nerve. But in fact, it was debunked. And how was it debunked? Through natural history studies that were conducted by my colleagues and myself. And that was critical to let us know that we were going to measure the wrong thing. We thought it was the nerve in the eye rather than the cortex involved. And so this helped design the very first in-human gene therapy research study for Canavan disease that was just launched by another company. Next slide. So this goes back to overall Ultragenyx's approach is patient-focused drug development. Drug development can be divided into 4 stages: the preclinical stage, early stage of development, late stage, and commercial. At every stage, we aim to address the following. We look to the individual and the family unit. We ask the questions: what is it like to live with this disease? What are your goals for treatment of your child? And how do they guide decisions about care and management? And what are your perspectives on the current treatment? Are they effective? What are potential treatments out there that you can envision? And what are the unmet needs that still exist? From the community, we look to the community that supports our patients and families. What existing supports exist? What are their influences for those individuals with or who are at risk of the disease? And what are the sources of information out there about the patient experience? What's out there, what's already been published, what's already been reported on, and how can we use that to apply to our research inquiry and to advance the whole field, to improve health outcomes of our patients. And so Ultragenyx involves patients all along the way to help us inform decisions about creating this about clinical development programs. So what do we most want to know, and what sources and methods are most appropriate are the main questions. Next slide. So here's a framework that we've taken from the Biotech Innovation Organization that shows that the patient experience data is really important throughout the product life cycle, including in early development. So in the research and discovery phase and leading up to preclinical development, it's really important to get patient experience information to start to inform the clinical development program. And it really guides our not only our clinical development, but our regulatory strategy, and it starts very early. Next slide. So currently, if we look at the stages of drug development, starting from -2 through 4, I'm just giving you an update of where we are at with 055. And we are at the pre-IND stage. So this stage is after we've initiated studies in vitro, which means in the Petri dish in cellular levels. In vivo means in live animal models. And then pre-IND is that continuing phase. And now we're preparing for regulatory guidance and starting to think about going towards the clinic. But what we're doing right now is advancing these 4 key areas of research. So we continue to make progress in our non-clinical or preclinical stage. We start to think about the manufacturing stage, and we'll go into more detail in the subsequent slides, the clinical stage and regulatory. And I'll give you an update of where we are in each area. Next slide. And this all occurs well before we're actually in the clinic. So in the preclinical or non-clinical stage, we are looking at or have looked at mouse models of CDD. We've partnered with other researchers to understand whether the gene therapy administered to slightly older mice might still ameliorate disease phenotypes. We learned that the AAV9 delivery of human CDKL5 to neurons in CDD mice lead to improvements across several different functional domains, but it didn't completely see improvement in other domains. So this helped us to start tweaking our preclinical program. Then we move on and start to evaluate the biodistribution in larger animal brains, and we vary the conditions using non-human primates and pigs. This is so that we can understand how many neurons do we really need to target to see efficacy with gene therapy. And this is a really important stage in preclinical development. We also start to evaluate and understand the biodistribution of the gene therapy and the toxicology, so the safety in studies that are considered good laboratory practice. So these are really rigorous studies that have high standards. And we're not just looking for observations of potential signals. We really need to have rigorous studies to understand where we express the CDKL5 human gene with this therapy and assess the initial safety profiles in animal models, okay? While we're doing that, next slide, in this pre-IND 0 stage, we start to think about manufacturing. And this is really interesting because as a clinician, I was always concerned about getting drug to the patient immediately. And what are the barriers from getting it from the animal stage to human consumption? And so we need to start thinking about making drug, actually making enough drug to administer in these larger animal studies. So we have to scale up, start producing more vector. This is not necessarily a very easy job. It takes time. We need to supply material that's called good manufacturing practice drug. So this drug has to be safe and effective for future clinical studies, and this takes time. Why is manufacturing important? Well, we need to make a safe and effective product. We need to generate enough virus for clinical trials to serve larger animals and then eventually humans, right, for clinical trials. We must comply with regulatory standards for safety and potency. It's very strictly controlled to make sure that we take every precaution as we make human grade material. Ultragenyx is actually in the midst of building our own manufacturing plant, so we can also address scalability and purification internally. And these are two key considerations when we're thinking about the manufacturing process. So this is also occurring at this pre-IND stage. Next slide. So from a clinical perspective, what are we doing right now? As I told you before, we engage with patient groups and clinicians very early, all the way back in -2 stage. And we continue to glean insights from caregivers, patient groups, and the clinicians. We start to think about what kind of study protocol, what's the overall clinical development plan. We want to understand the diagnostic journey, the burden of illness, addressing all the various parts of the illness and not just one key feature such as seizures. We want to look at everything. Where's the unmet need? What's the ideal treatment in the eyes of both the caregiver and also of the clinician, the experts that are managing the patients? What are barriers to treatment? This is a CNS disease. We need to target the brain. What do we have to think about with gene therapy and be concerned about to get to the brain? We start to think about the assessments that we need to measure within a short time frame. Clinical trials are usually 1 to 2 years, sometimes longer, but we have to see what are we going to do as far as measuring change over time. We also want to think about dose administration. How do we get the gene therapy to the patient effectively and get the biodistribution that we want based on our preclinical information? And also about recruitment. Where are patients located? How are they being diagnosed? Can we find them? Can we get to all patients? We also start conducting literature reviews, understanding what's been published, right? So we want to understand what is known and accepted. We also conducted retrospective chart reviews with Ultragenyx to understand, is this compatible with what's published? Are there other features of the disease that we don't know about? We start to initiate biomarker development, and we'll talk a little bit more about that. Looking for fluid or non-fluid markers that tell us about the disease outside of the clinical features, something that we can measure potentially early and see if it's responsive to change over time. We have to start to investigate this now. We also are supporting external efforts. So the CANDID study, we are actually financially supporting. We also support and encourage our UO1 NIH grant colleagues who are working on that grant, and we sit on advisory boards for them as well. So that's happening from a clinical. The fourth area on the next slide is about, and then we address regulatory. So how is the CDD community helping us to Ultragenyx advance our program? And it's through observational clinical research. So I mentioned the Allstripe study to explore how CDD manifests over time through this retrospective chart review. How do patients interact with this health system? What are the types of visits? What's the burden? And we validated existing knowledge that has been published. So in this area, we realized that the features of seizures and cognition and language and visual impairment are what we thought. And so that helped us to verify and allow us to chart our course forward. Biomarker development, we're going to start exploring potential markers of CDD to allow us, as I said, to potentially see an earlier response to a disease-modifying therapy. We're also patient-focused research. So we had initial insights meetings with US and Canada to increase understanding of our perspectives on the treatment that is being currently offered and the unmet needs. We also explored knowledge of and perspectives on gene therapy from a cohort of caregivers, conducting one-on-one interviews with caregivers in the US to get a more detailed exploration of symptoms and the impact on the family and the patient, build on existing data from these internal and external CDD research efforts, and hopefully inform long-term endpoint development. Our second insights meeting for US and outside of US is to understand the perspective and needs regarding participation in an early stage gene therapy. Would families want to engage in gene therapy? Is this the approach that caregivers are amenable to? What kind of support would a family and patient need to participate in a gene therapy trial? And this will help shape our clinical trial informed consent process and help us support you when we're trying to execute these potential trials in the future. So this preclinical research and future clinical, so we gather these insights from patients and caregivers, and they're directly informing our clinical development program. Next slide, please. So additional examples are advocacy groups and caregivers. We're really learning from these initial insights meetings and regular engagement with the community through our patient advocacy groups and interactions with global clinical development. You know, we're learning about the most important symptoms to treat across age groups and males and females, all those domains, and the ability to stop other medications, impact on seizures, cognition, motor, and vision. You know, many consider gene therapy to be their major hope for treating CDD because they'll feel like it targets the root cause. So these are things that we've already discovered and verified with the patient community. We want to minimize study burden wherever possible, right? Patients who have multiple healthcare visits to just manage their day-to-day, adding a trial participation can add significant burden. How can we lessen that burden? And it's really important. It reinforces that we have to have timely communications with the community so that you understand where we're at and what we're doing. And there's an opportunity to continuously learn from the global advocacy groups. So we don't want to be focused on just one country or one experience, right? How does CDD affect caregivers and patients in other countries? What are the knowledge gaps there? Are there different perspectives on unmet needs? We can't assume one vision based on one community. We have to think about the global community as well. Click on the next. We also interact with what we call KOLs, so key opinion leaders. So I was a KOL in academia on lysosomal storage and other neurogenetic and epilepsy disorders. And so here, the same. We engage expert physicians and healthcare providers. They might be an expert in a particular disease state. They actually might also be, may not be in the particular disease state, but maybe in gene therapy. So we're looking at the entire picture. We're looking at the disease state, engaging our key clinicians who know about the clinical manifestations of CDD, but we're also engaging the top minds in gene therapy to understand how can we bring gene therapy to the brain. So they're really playing a critical role at different time points to make sure that we are getting an accurate picture. So disease expertise, gene therapy expertise, even expertise in study design, eligibility criteria, endpoint strategy. So we will engage people who are specialists in certain endpoints and certain aspects. And we also collaborate with other researchers. It's really important for Ultragenyx to communicate back and forth. We have a commitment to be transparent, and what we learn, we want to share back to the community. And we also support these external efforts. The Lulu Foundation has put out a CANDID study, it's called CANDID, looking at endpoints. And Ultragenyx is one of 7 companies supporting it financially and with those of us sitting and helping guide and answer questions and help with the execution. UO1 grant, so Ultragenyx has 4 representatives that were invited as members of the External Advisory Committee, and we support and engage with your respective researchers. It's really important because if we can help push forward research in one area, it benefits everyone. Next slide. So what are the current opportunities for our CDD community to participate in Ultragenyx research? And we're really appreciative of IFCR to share information and updates from Ultragenyx to the CDD community about our following research efforts. So right now we're conducting one-on-one interviews. These are open to caregivers who are living in the US and fluent in English. If you'd like to learn more, here's a contact right here, Natasha Schumacher. There's the email and phone number. We are having that second Insights meeting, and this is open again to caregivers living in US, Canada, UK, Germany, Ireland, Netherlands, Italy, and must be fluent in English for this particular aspect. And here's some contact information. So this is how you can continue to contribute and help us understand your particular concerns about CDD. Okay, next slide. So biomarker development, I wanted to spend a minute or two on this. So biomarkers can be blood markers, it can be other fluid markers, it could be a functional assessment, electrophysiology, it could be imaging. Biomarker development is not novel to CDD. It's an exploration for most rare diseases, and we're looking for some kind of signal of the disease that is not a clinical symptom. So what is the value of a biomarker? It can actually accelerate drug development. And that's because if it's an early marker, we could start to see that a therapy is working or that there's a change with different dosing. So there's a critical role if we can identify a valid biomarker of CDD. So it could serve as that early signal that we're responding to treatment. It can inform the design of investigational trials and help us with the selection of a dose. And it can contribute to the overall regulatory approval. So it's not just on functional outcomes, but if we're showing a biochemical change or another change that is representative of CDD, then it could help us with our application for approval. But the problem is development of a biomarker related to brain function is very challenging. And so it's really important to support all the efforts out there for biomarker research in CDD. Next click. So multiple innovative approaches are necessary. We don't look at biomarker development as a competition. If we think of it as, we want to support the researchers any of the researchers who are investigating various avenues. One way is not necessarily better than the other. We do not know of a biomarker in CDD yet. So we're encouraging future efforts, including the pending biorepository supported by Lulu, that's being developed by researchers at the UO1 NIH study. We, Ultragenyx, are planning to explore potential biomarkers. That is part of our normal clinical development programs for each of our programs at Ultragenyx. And whatever we learn about the biomarker, whether things work or don't work, we want to share back. So our analyses, we will share back with the community so that we can avoid waste of time, effort, and resources. You know, you can imagine if we explore one marker and it doesn't work, then we want to inform others and say, well, you might want to investigate it or you might want to alter it. It's informative. Even negative results are important. So we can start to look at other potential candidates. And we anticipate that these results will inform other stakeholders. So that'll help others push the whole field forward. Okay, so more to come on that topic in the future. Next slide. So the last few minutes, so we talked about the clinical stages of the pre-IND or stage 0. But just a comment on regulatory. While we're thinking about the preclinical development, understanding whether a gene therapy is working in animal models and is it safe in the animals, then we're thinking about how engaging in the clinic, are we going down the right path? We have to start thinking about regulatory interactions. And I'm not sure how familiar you are with this process, but every step of the way, we're interacting with regulatory bodies such as the FDA. They have these interactions really guide our clinical development plan and our future IND submission. So investigational new drug submission. We have to apply to even think about testing this in humans. And before that, we have to conduct a variety of pre-IND and scientific advice meetings with these health authorities. And there's a back and forth, a communication to say what should and shouldn't be done, what should we think of. So they're informing us as well. It's not a one-way communication. And so it's an iterative process. We start to come up with things, then we have a back and forth. And it can be common that we have different perspectives from different health authorities. So what the FDA says versus what happens in Europe might be different. And we ultimately must seek approval from a health authority in order to even conduct a first-in-human clinical trial. So all of this work is happening well before you see a study open for a clinical trial, okay? So these are all the activities that are happening currently at Ultragenyx regarding CDD. Okay, next. So looking to the future. So we're preparing in clinical development. We have not yet identified specific eligibility criteria for any future CDD trials. It's really important that our overall goal, our overarching approach is to study gene therapy in a broad patient population that reflects the predominant features of CDD. Again, this is going to occur during a pre-specified timeframe, right? These trials are not unending or never-ending. It's a timeframe that is also informed and given feedback by the FDA or other authorities. We have to look for the change in symptoms and the disease manifestations to see how to measure them so that we can say whether or not a therapy is modifying the disease course. And it's really critical that we understand that families understand the responsibilities of participation in these potential trials and the impact. So we're going to talk with families and inform them that, we want to ensure that studies are feasible, that they're not so difficult and complex, and understand if families have the time and the bandwidth to take it on. There's also risk, right? For first-in-human gene therapy, it means that it's never been tried in another human before. And so is this a risk a family is willing to take? Early stage trials are focused on safety and dose finding and not efficacy. And gene therapy is a one-time treatment currently. We are not able to redose in the world of gene therapy at this time. So that's another consideration when you're thinking about participation. Understanding risk, understanding irreversibility, or at least that it's a one-time, it's not something that is recurrent. So, and clinical trial participation requires frequent visits to understand and assess the key features of CDD, in addition to your standard of care health maintenance visits. And so it is a huge burden for families. So we want to think about this thoughtfully. We want to try to reduce burden while maintaining the rigor and integrity of a clinical research trial. Next. Oops, I lost. Oh, there we go. I want to comment on durability of a gene therapy. It's a really important consideration. It was a great question. So what do we know about redosing and durability? Well, when we think about durability, it's the length of time that a patient will experience a therapeutic effect from the gene therapy. Is it going to still be effective as it was from day one, 5 years, 10 years, 20 years later? And researchers and scientists are working to understand if we can redose to extend durability if there's a concern that there's a loss of durability. And is it feasible? Is it safe? With gene therapy, we think about potential safety issues if we try to redose. At this time, there have not been any FDA-approved gene therapies that have been delivered more than once to a patient. And so we have to understand what are the limits and what can we do. In general, durability, when we think about the central nervous system or the brain, durability targets organs, and if they're actively dividing, then we think about a concern for durability. For neurons, though, if we're targeting neurons in the brain, humans are born with the majority of neurons that you'll ever have in a lifespan. There's very little turnover of new neurons. So we don't expect, but we have to confirm this, that we'll lose the transgene once we get the gene into the cell. And then hopefully, therefore, that transcript will be expressed for that life of that neuron. So maybe the brain will prove more durable than other organs that are actively dividing because we're not diluting or reducing that gene transcript over time. It's a very complex thought, but we have to test it out. So that's why you'll see follow-up of gene therapy programs are for years after. Even though it's a one-time injection, we see patients in other gene therapy programs coming back yearly for years to see safety and efficacy. And so that's a question that remains to be seen. But we're hopeful if we can target a neuron that we will have long-term expression. That's our goal. Next question, or next slide. So other resources about the drug development process, our patient advocacy team, Kristen, who you know we work with in patient advocacy, have developed a website to address these topics you're most interested in. The link is right here to learn more about clinical trials in general and the drug development process. Finally, resources about gene therapy. Again, we have resources specifically about gene therapy on this link provided here. And the American Society for Cell and Gene Therapy is also a great resource for patients and caregivers. And you can click on this link, patienteducation.asgct.org. It's a fantastic resource. There is a Spanish version for gene therapy in you available as well. Okay. All right, so thank you so much for your time and listening. And now we'll move on to the Q&A.
Thank you so much, Heather. This has been so informative, and I know that the community is going to appreciate this. So I'm going to jump on in and ask some of these questions here that we have. What is the estimated timeline for clinical trials to start for your gene therapy?
So as we discussed, we're currently in the preclinical phase of drug development, stage 0, and working towards a future investigational new drug submission, the IND that I talked about. The preclinical studies include conducting animal studies, and this phase of research really needs to be completed before any clinical trials are initiated studying the investigational treatment in patients. An IND is required before any clinical trials can be conducted, and it's very common for preclinical work to take several years to complete because of the extensive testing that's expected and required by these regulatory agencies. It's also important to note that because we're still in the preclinical stage of research and development, it's important that to realize that it's possible that our work may not successfully lead to a clinical trial. So I have to caveat that. Ultimately, though, if our preclinical work is successful and we are able to advance to conducting clinical trials in patients, we will work with the regulatory agencies to conduct these trials in a regulated and safe way. We'll incorporate their feedback to make sure we have designed studies with safety in mind. And that allows us to collect all the important information to determine how effective and safe the drug is at addressing the disease. We understand that the community is eager to learn more, and we will share a timeline with you with when the OAK55 program has advanced to the next stage. So our team is working with a sense of purpose and urgency to advance this research as quickly as possible, and we will come back and give you another update. We have more to tell.
Okay, thanks. And this is kind of a batch of questions I'm going to read through. It's a mix of questions that were focused on eligibility criteria. So what is your ideal patient type? Who will be eligible? If you don't know yet, when will you know? And are there any previous surgeries, especially a corpus callosotomy, or any previous trials that would exempt us from the gene therapy trials? Any age or gender or medication exemptions? What procedures or treatments available right now can potentially affect gene therapy, like growth attenuation therapy or even VNS?
So, as we're currently in the preclinical phase of drug development, we have not yet developed a clinical study protocol. So the clinical study protocol will define eligibility criteria such as age, sex, what existing treatments or medical history we can exclude from participating, but we do not have a study just yet. Our work is at that stage. We're conducting preclinical studies and we're learning. So we are learning about CDD from patients, caregivers, and expert healthcare providers to understand and help us define this. So as we work towards that future IND submission, we will incorporate feedback, this critical feedback from you and from the community that's participating in all of our interactions, and as well as our physicians and researchers, will be incorporated into the design of the clinical trial and its study protocol.
Okay. Will people in the UK have access to clinical trials? Is this on a different timeline?
Right. So we have not yet identified where we would conduct a CDD clinical study. It's important to understand that the geographic distribution of patients living with CDD is really important when we're planning for clinical trials. So we're going to take in as much information as possible. And we have to understand the local regulatory agencies' requirements in these individual countries. And then we can ascertain whether it's feasible or not. All these factors will help determine where the clinical trials are conducted. Generally, Ultragenyx's goal is to develop therapies with the potential to treat as many rare disease patients as possible. And this typically requires that we conduct clinical trials in multiple countries. It is common for most clinical development programs to begin in a smaller number of countries and then expand into larger countries. And that's what I can say at this time.
Okay, and you've kind of covered this one, but the next question is, once gene therapy is available, which country do you think will be the first to approve it?
Right, so this is talking about approval. So it's important to highlight that we do not know whether gene therapy will ultimately be available. You understand, we don't know this at this time because we haven't conducted any clinical trials in patients living with CDD. We do recommend that you keep in contact with IFCR, who can provide you resources on the stages of drug discovery and development, which will be helpful in understanding the different phases. So listen to the phase and the length of time of drug development. The ASGCT that I highlighted already has a lot of information, tools and programs for patients, caregivers, and organizations to understand that drug development process.
Okay. What kind of cognitive improvement are you measuring success for? Will gene therapy succeed to stopping seizures or just mitigating?
Okay. Once again, it's important that we acknowledge that we haven't studied gene therapy in humans, right? So this is all preclinical information, and we're in the preclinical phase. So we don't know how well a person might respond to gene therapy because we haven't conducted the studies yet. So as we're currently in the preclinical phase of drug development and we're not yet developed a clinical protocol or which measures, right? So our work at this stage is conducting preclinical studies and learnings. This goes back to learning from the caregivers and expert healthcare providers about these cognitive measures. Examples of the learning activities. So, again, conducting the interviews and insights. I can't tell you how critical it is to talk directly to you, the parents, to understand their perspectives on the most important symptoms so that we're targeting the right symptoms and aspects of the disease. We also are really supporting and in multiple ways the CANDID and YO1 studies to help accelerate this knowledge of these potential clinical assessments. We're listening is the most important point. We're listening to you all.
What do I, as a parent, need to do to help bring gene therapy for CDKL5 to a clinical trial?
That is a wonderful question. It's an important question. And thank you for asking. So people living with rare diseases and their caregivers and families, they play an instrumental role in helping to advance drug development. Patient advocacy is important. We recommend that you contact IFCR to learn about the specific research opportunities that are ongoing. For example, you might consider learning about two efforts designed to inform the selection of potential assessments, right? So I talked a little bit about the current, the potential CDD clinical trials, the CANDID trial and the YO1 studies. So participation in those studies are informative. Going back to what we are offering at Ultragenyx right now, are opportunities for engagement with caregivers that will help us. We want to hear your perspective, and that's going to inform our clinical development program. These are the one-on-one interviews that are open to caregivers living in the US and fluent in English, and the international insights meeting, talking to caregivers in other countries. Right now it's US, Canada, UK, Germany, Ireland, Netherlands, Italy, and also fluency. So those are some opportunities, to engage in research. When you participate in one, it helps advance the whole program, the entire community, scientific community forward.
Right. For all of you listening, that's our Be One of 200 campaign, and then the CANDID study. There really is great reasons to be involved in those things. So thank you for pointing that out, Dr. Lau. If you do gene therapy once, does this prevent you from doing it again if it doesn't work, or either with your treatment or with someone else's?
That's a good question. It's important to acknowledge again that we haven't studied gene therapy in humans. We're still in the preclinical phase. So obviously that's important to highlight again. But at this time, we expect that gene therapy will be a one-time treatment, and that because there are not any FDA-approved gene therapies that are serving the brain or other areas that have been delivered more than once to a patient. So at this time, this is what our current abilities are. Okay, so I think those are the submitted questions, and try to address them during the actual presentation. So it's important to know, as from Ultragenyx, that it really helps to hear from the community. It helps to hear these questions so that we can deliver answers. We have to we sit down, thoughtfully review the questions to answer them to the best of our ability. So it's a little bit difficult, but I appreciate this opportunity to be live and discuss it with you.
Yes, and thank you so much for sharing this and for sharing the opportunities where parents and caregivers can use their experience to help you all learn. And that's it. To me, it's exciting and it's encouraging that as a company, you all are starting there. There's a lot of work to do, and we're excited that people are doing it and keeping the patient community at the center of that. So thank you so much for your time and effort to educate the community. We really appreciate it.
Thank you for this opportunity, and I appreciate the engagement between IFCR and our patient advocacy group. Kristen Voorhees is our representative, and the continued learning. I learn from you all every time I speak with you. Our ears are, we're listening all the time, and it's really important to gather information. You never know what will be the critical piece that will kind of spark something and help us with our overall approach. And I think, again, it's important to highlight that Dr. Kakis and Ultragenyx is committed to sharing. So when we can, we like to share information back, our learnings. We want to share that back to you. And it might be relevant for others in the field as well.
Okay, and to the IFCR community, CDKL5 community, thank you for watching. And we will continue. Oh, and you can submit questions like Dr. Lau said, keep communication with us open. If you have questions, submit them and we can relay them over. And as they can answer them, they will. Keep in mind, as you heard, sometimes there's questions they just can't answer yet. It's not because they don't want to. There's regulatory reasons and all types of reasons, but keep those communication with us open, and we will continue to help our community's cause be heard.
Yeah.
Thank you all so much.
Thank you.